Hello, everyone. So we are going to talk about TCF4 related neurodevelopmental disorders as part of GenomeArc case series. And this is an ongoing case series that we will discuss different complex genetic cases with different mutations and their clinical presentation. So this is a clinical presentation of a proband who is 2.5 years old. And the onset of the clinical presentation was around 8 months. And the key features for her clinical presentation includes developmental delay. She was unable to walk, stands only with supports, no clear speech at this age. She had poor understandings of her surroundings. There are mild hyperactivity also observed. Stereotyped midline head movement has been observed, and she have a very poor weight gain. And since it's a girl, the initial suspicion was maybe it's something to do with Rett syndrome. So the clinician was forwarded this patient to the genetic lab for Rett syndrome screening. And they conducted, for the diagnostic test, they conducted exome sequencing using MGI sequencer, DNB-seq G50 that targets around 60 to 70 megabase of human coding regions. That covers almost 100% of the protein coding genes. So it's a very high coverage kit for exome sequencing. And exome sequencing is the first year test for this kind of neurodevelopmental disorder cases, because there's a lot of these variants that have been associated with genetic disease. And more on that, so this will exclude all the CNV detection, because this test will only look into whole exome sequencing data. So when we sequence it MGI, it gives you about, almost 80 to 100,000 variants, depending on the samples. And from then onwards, you need to identify if there is any causal variance that can be attributed to the clinical presentation that we see in this girl. So basically, this case, when this case was presented, the patient blood, you go through sequencing, as we showed. The secondary analysis is based on the MGI platform, but we used GenomeArc Horizon analysis. Is a tertiary platform that can do rare disease, diagnosis based on SEMG, cancer diagnostics based on AMP, and it also can do target detection for drug design and predictive medicine. So our aim is to identify the variants using GenomeArc Horizon analysis, and the aim is to find the germline variant, since this is blood DNA, germline variants from this rare disease module within the GenomeArc Horizon platform. So this is the findings from the GenomeArc Horizon platform. These findings actually was identified after uploading the VCF file into the software, and it identifies a lot of pathogenic and likely pathogenic variants. So roughly around 40 of these variants, but a lot of these variants are not related to the symptoms, and some of these variants are actually sometimes have low quality, like low heterozygous quality listed, as you can see in the table. So maybe false calls too. So when we looked at this data, we identified this variant TCF4, impacting TCF4 gene, and this gene being known for this type of clinical presentation. And I will explain a little bit more how we analyze the data from GenomeArc Horizon platform. So first, we look at the population frequency of the variant. So you can see the variant table. After uploading the VCF file, it automatically identified classify the variants based on SMG, and gives you the short list of clinically meaningful variants. So this is a stop gain variant, TCF4, that has no population frequency in NOMAD, and also GenomeArc Horizon have its own control frequency from different studies that they collaborated on. So this platform shows this variant is ultra rare variant. Then it also shows that even this population is ultra rare. There are 3 pathogenic samples being reported in ClinVar. And these are all pathogenic variants. That's pretty assuring for this type of cases. So we then looked at what kind of in silico prediction for the variants. It seems like the CAD is pretty high, where the CAD kind of ranges from 20 to 25. Seems to be deleterious. It has a 39 score, and the inheritance of this variant is autosomal dominant, because the zygosity, if you look at the zygosity, it's actually a heterozygous variant, and it kind of fits the picture and predicted to be likely pathogenic or pathogenic in this case. Okay, so this platform gives you a holistic view of this data that gives you enough information to make a decision. And we looked at the variant exon, just to confirm, and also the transcript within Horizon platform. And this is the report that's been generated with pathogenic and also the SCMG code right next to it during the classification that can be printed. So this report is auto-generated from the Horizon platform. So it's basically premature stop codon that truncates the most of the protein after exon 18. So there's a lot of exon that will be missing. Okay, this is not exon 18, this is exon 10. So there'll be a lot of sequence that will be missed after exon 18. So yeah, the supporting evidence according to GenomeArc Horizon platform are PVS1 because it's a loss of function variant, PM2 because it's absolutely absent in the population, PP4 because patient phenotype highly specific to TCF4, and also there are other classification supporting evidence goes with ACMG guideline. For example, that we have additional 3 cases been reported for this variant. All those 3 cases have similar kind of phenotype. So the classification of pathogenic makes sense. And the gene relevance, for this particular patient and for neurodevelopmental disorder is very important. So TCF4 gene encodes a basic helix loop helix transcription factor that serves as a master regulator of gene expression during embryonic development. And it functions by binding into, so it's a DNA binding protein that actually kind of, regulates other genes. And it actually binds to a known sequence called Eprusi boxes, characterized by the consensus sequence CANMTG. So it binds into that particular, sequence identity, and then it regulates downstream targets, whether it's a gene or other functional elements. So having a premature stop codon will have impact on the binding, the DNA binding affinity of this protein, because the protein will be severely truncated as it is prematurely mutated at exon 10. And because TCF4 is widely expressed throughout the brain, it is absolutely critical for healthy nervous system development, including the proper differentiation and migration of neurons. That's been reported in numerous publications. So you can see kind of like a very supporting evidence of having a loss of function mutation impacting TCF4 can impact the brain development. And also TCF4 TCF4 known to be correlated with Pitt-Hopkins syndrome. So Pitt-Hopkins syndrome, the core characteristics actually are intellectual impairment and autism spectrum features, delay motor and speech development, distinct facial features, and hyperventilation, stereotypic hand movement. So this is a lot of these observation for Pitt Hopkins, clinical phenotype we also see in our case. So maybe there are few additional observation in our cases, but a lot of these overlaps. And the known gene for this Pitt Hopkins syndrome is TCF4. So TCF4 variants actually, there are cases accumulating with different types of variants. So we have observed in the literature there are 6 cases being reported. Out of these 6 cases, we found 3 of these variants are de novo. And interestingly, even though our case is female, this gene is not exclusive to, or this condition is not exclusive to female. We saw out of these 6 cases, 3 are female and 3 are male. And all of the 4 patients that we know age of, they're all early onset patient. They presented their, they presented their phenotype very early on. And interestingly, all 4 that we saw that have the phenotype there's a lot of overlapping phenotypes, but not necessarily unique. Similar. So all 4 of these patients that we have detailed phenotype or clinical information of, we can see that there is a lot of overlapping features, but there are also unique phenotype that attributes to what type of mutation, where it is in the gene that gets mutated, and also the genomic background that contributes into the phenotype. And also, so some of these patients went through some other tests as well, but those tests came out negative, like for example, FMR expansion or some, MACP2 sequencing, because some of these patients are suspected to be Rett syndrome, but it came out negative. So basically, applied therapeutic and management includes a multidisciplinary approach. There is no drug at this moment, but behavioral therapy and developmental support is very important because they present their clinical presentation very early on. And a regular follow-up with the hospital or clinic is super important, because as the disease progresses, the intervention might need to take place, and it can be intervention of different type of therapy, communication support, or there are different types of ABA therapies out there. So those therapy can be integrated very early on for a better outcome. And as I said, there is no drugs at this moment for TCF4 related syndrome, but the future genomic medicine looks very promising because those technology are designed to target specific mutations. So antisense oligonucleotide and small interference RNA, siRNA, these 2 technologies are very promising that actually targets this kind of specific mutation. And in future, hopefully, there will be, ASO or siRNA drug that can be designed for these TCF4 related mutation and syndromes that can reverse the course of the clinical presentation. So that is the hope for precision medicine for not just TCF4, for many different rare diseases and rare neurodevelopmental disorders. So thank you for joining the case series, and we are hoping to bring more of our interesting cases that we identify interesting variants through Horizon platform, and we'll be happy to take any questions during this webinar. Other than that, please stay tuned. We'll be bringing more of this case series towards you.